Hi everyone and welcome. I'm kicking off a new series of videos which are going to be in a podcast style, hopefully educational podcast like videos. So please stay tuned for this, and if you learn something from this video, please give it a thumbs up so other people can learn about it too.
All right, welcome back everyone to the deep dive. We're diving into the microscopic world today, focusing on something pretty vital to, well, all life as we know it.
That's right. We're talking about mitochondria, often called the powerhouses of the cell.
Powerhouses, exactly. And, for a long time, I think we've all kind of pictured them as these, static bean-shaped things just kind of hanging out in the cell.
Yeah, like from those old textbook diagrams, right?
Yeah.
But the reality is so much more dynamic.
Oh, absolutely. So today we're really going to unpack how scientists are using some pretty nifty tools, fluorescent dyes, to actually see these tiny powerhouses in action. And importantly, how their shape, their morphology, can tell us a whole lot about what's going on inside a cell.
And that morphology, it's not static, right? It's constantly changing, adapting. That's where things get really interesting. And also, to be honest, where the challenges start to pop up.
Exactly. We've got a ton of really cool research papers to dig through today. And our mission is to really understand how these dyes work, what they can reveal about mitochondrial morphology, and just as importantly, all the things that researchers have to consider to make sure they're getting the right picture.
Because it's not always as simple as just adding a dye and looking under the microscope.
Right. So to lay the groundwork here, we know there are different types of fluorescent dyes out there. Some are designed to target specific parts of the mitochondria. Some are used for tracking movement. And others can tell us about things like membrane potential or reactive oxygen species.
Yeah, all those functional aspects. But today, our focus is really going to be on the dyes that are primarily used to see the structure, the shape of the mitochondria.
So those morphology dyes, but just to be clear for our listeners, we might touch on some of those other types of dyes because, as you mentioned, function and form are often intertwined.
Absolutely. You can't really separate them completely.
Okay, so let's jump right into one of the first big challenges that researchers face when using these dyes to look at morphology. And it has to do with this concept of mitochondrial membrane potential, or M.
Yeah, M. It's kind of like a tiny battery inside each mitochondria.
A battery. Okay, so walk us through that. How does a battery come into play here?
So the inner membrane of the mitochondrion is actually electrically charged. It's got a negative charge on the inside and a positive charge on the outside. And this difference in electrical potential, that's the DEM, is absolutely essential for the mitochondria to do their job, which is, of course, producing energy for the cell.
Right, making that ATP. It's like the currency of energy in our cells.
Exactly. Now, here's the thing. A lot of these fluorescent dyes are actually drawn into the mitochondria because of this membrane potential. They're attracted to that negative charge inside.
So the dye is like, ooh, negative charge, let me go hang out in there.
Pretty much. And that's great because it allows the dye to accumulate specifically in the mitochondria, making them light up so we can see them. But, and this is a big but, it also means that if something happens to change that membrane potential, the way the dye is distributed inside the mitochondria could change too.
Ah, so even if the physical shape of the mitochondria hasn't actually changed, it might appear different under the microscope simply because the membrane potential has fluctuated, and that's messed with how much dye is in there.
Exactly. So researchers have to be very careful when interpreting the images. They need to consider whether any changes in morphology they see might actually be due to changes in membrane potential rather than actual structural changes to the mitochondria themselves.
Okay, so that's a pretty big thing to keep in mind, right?
Yeah.
You might think you're seeing fragmentation or elongation of the mitochondria, but it could just be a change in that electrical charge messing things up.
Right, and that's why it's so important to understand the mechanisms of these dyes, how they get into the mitochondria, and what factors might influence their distribution.
Makes sense. Now, it gets even trickier because some of these dyes can actually cause changes in the very morphology they're supposed to be helping us observe.
Oh yeah, that's a real concern. Certain dyes, if used at too high of a concentration, can actually have unintended side effects on the cell, and specifically on the mitochondria.
So they can kind of mess things up themselves.
Exactly. One example that comes up in our sources is Rhodamine 123. It's a dye that's often used to measure membrane potential, but there's evidence that it can actually induce changes in mitochondrial shape. So if you're not careful, you might be looking at a morphology that's been altered by the dye itself, rather than the true native state of the mitochondria in the cell.
That's almost like, I don't know, using a really heavy hammer to tap on a delicate sculpture to see if it's hollow.
Yeah, that's a good analogy. The tool itself can influence what you're trying to observe. So researchers have to use these dyes with extreme care, often at very low concentrations, and be very mindful of potential side effects.
It's like a constant balancing act, right? You need enough dye to actually see the mitochondria clearly, but not so much that it starts messing with their structure or function.
Exactly. And it really highlights the importance of careful experimental design and having appropriate controls to make sure you're seeing what you think you're seeing.
So it's not just about pretty pictures. There's a whole lot of scientific rigor that has to go into these experiments.
Absolutely. You have to be critical, you have to understand the limitations of the tools, and you have to be able to interpret those fluorescent signals in the right context.
Okay, so we've talked about membrane potential. We've talked about dyes potentially causing changes themselves. What are some of the other challenges that researchers have to consider when looking at mitochondrial morphology with these dyes?
Well, another important factor is that the brightness of the fluorescent signal you see under the microscope isn't always a direct reflection of the size or shape of the mitochondria.
Okay, so why would the signal be brighter or dimmer if the morphology is the same?
It can depend on a few things. For example, the total amount of mitochondrial material within a cell can affect the overall brightness. If a cell has a lot of mitochondria, you're going to see a brighter signal overall, even if the individual mitochondria themselves aren't any bigger or differently shaped.
So it's like having more tiny light bulbs packed into the same space. It'll look brighter overall.
Exactly. And then, of course, the concentration of the dye itself can also play a role. If you use a higher concentration of dye, you'll generally see a brighter signal. But again, you have to be careful about those potential side effects we talked about earlier.
Right, too much dye can start causing problems, so it's another thing that researchers have to control for very carefully in their experiments.
Absolutely. They need to make sure they're comparing apples to apples, so to speak. If they're looking at changes in morphology between different experimental conditions, they need to be sure that any differences in fluorescence intensity are truly due to changes in morphology and not just variations in dye uptake or the amount of mitochondria present.
So again, it's all about careful interpretation of the data and taking all these potential confounding factors into account. Now, another practical aspect that seems important is whether a particular dye can be used in cells that have been fixed, right? Like chemically preserved.
Yeah, that's a huge practical consideration that really influences how you design your experiment. Some dyes are fixable, meaning that they stay put inside the mitochondria even after you treat the cells with chemicals to essentially freeze them in time.
And why would you want to fix the cells?
Well, it's often necessary if you want to combine your mitochondrial morphology studies with other techniques. Like, let's say you want to visualize specific proteins within the cell using antibodies, which is called immunostaining. Or maybe you want to look at the genetic material, the DNA or RNA, using a technique called ABPH.
Right, those techniques often require fixed cells because they involve a lot of steps that would damage live cells.
Exactly. So if you want to do those things and also see where the mitochondria are, you need a dye that can withstand the fixation process. And some dyes are really good at that. They're very stable, and they stay brightly fluorescent even after fixation.
So those are the fixable dyes. But what about the ones that aren't fixable?
The non-fixable dyes, well, they're really only useful for looking at mitochondria in living cells. They'll wash out or fade away during the fixation process, so you can't use them if you need to preserve the cells for other downstream analyses.
Okay, so that's another big decision point for researchers, right?
Right.
Choosing the right dye for the job based on what they ultimately want to do with those cells.
Exactly. It's all about matching the tool to the research question.
All right, so that really sets the stage for us, all the challenges and things to consider when using these dyes. Now let's actually delve into some of the specific dyes that researchers use to look at mitochondrial morphology.
Sounds good. Let's get into the specifics.
So first up is MitoTracker Green FM. It's a classic, right?
Oh yeah, MitoTracker Green is a workhorse in the lab. It's super widely used for visualizing mitochondria in living cells, and its mechanism is pretty straightforward. It's a small molecule that can easily cross the cell membrane. And then remember that negative membrane potential we talked about?
The VAM, the battery.
Yep, that's the one. So MitoTracker Green is drawn into the mitochondria because of that negative charge. Once it's inside, it fluoresces bright green when you hit it with the right kind of light.
So it basically lights up those active mitochondria, letting researchers see their shape and how they're all connected within the cell. And this is all happening in real time, right? We're talking about live cells here.
Absolutely. It's like a live view into this dynamic world inside the cell.
Yeah.
You can actually watch those mitochondria moving around, fusing, dividing, all in real time.
Wow, that's incredible. But we know there are limitations, right? Especially when it comes to using it for morphology studies.
Right. The main limitation of MitoTracker Green is that it doesn't form a permanent bond with anything inside the mitochondria. It's just hanging out in there as long as that membrane potential is maintained.
So if that electrical charge fluctuates or disappears for some reason, the dye can just leak back out.
Exactly. It's like if you had a bucket with holes in it and you were trying to keep it full of water. If the pressure pushing the water into the bucket drops, the water's just going to leak out those holes.
Okay, I get the analogy. So if a cell is stressed or damaged and that mitochondrial membrane potential goes down, MitoTracker Green might not be the best choice for visualizing the morphology because it could give a misleading picture.
Exactly. And the other big limitation is that because it doesn't bind permanently, it doesn't survive the fixation process. If you try to fix the cells to preserve them, the dye washes out.
So you can't use it if you want to do those other techniques we talked about, like immunostaining.
Right. It's really best suited for taking snapshots of mitochondrial morphology in living cells where, the membrane potential is stable.
Okay, so it's a powerful tool for certain types of experiments, but it's not a one-size-fits-all solution.
Definitely not.
All right, so let's move on to another popular dye, MitoTracker Red CMX Rose. This one sounds a bit more complex with that chloromethyl group in its name.
Yeah, so MitoTracker Red, along with some of its cousins like Orange CMTM Rose and Deep Red FM, they've got a really neat trick up their sleeve. Like MitoTracker Green, they initially get into the mitochondria because of that membrane potential. They're attracted to the negative charge.
So, same starting point.
But then that chloromethyl group comes into play. It's really reactive with certain types of chemical groups called thiols, which are often found on proteins.
Okay, getting a bit technical here, but basically it forms a strong bond with the proteins inside the mitochondria.
Exactly. It latches on and doesn't let go. And that makes it really useful for morphology studies because now, even if the membrane potential changes or the cell is stressed, the dye stays put.
Ah, so it's like it's anchored in place.
Exactly. And the other great thing is that this covalent bond, it can survive the fixation process. So you can fix the cells, preserve them, and the dye is still there, still brightly fluorescent, even after all those harsh chemical treatments.
That's fantastic for those experiments where you want to look at the mitochondria in relation to other things in the cell, right?
Exactly. You can do immunostaining to see where specific proteins are located. You can do FF to look at the genetic material, and you can still see those beautifully labeled mitochondria all in the same fixed sample.
So it's much more versatile than MitoTracker Green in that sense.
Absolutely. But of course, there's a trade-off. Once that dye is covalently bound, it's no longer sensitive to changes in membrane potential.
Right, because it's basically stuck there, whether the battery is charged or not.
Exactly.
Nice.
So if you want to measure dynamic changes in membrane potential, MitoTracker Red CMX Ross isn't the right tool.
Okay, so again, it's about choosing the right tool for the job.
Yep.
Now, we also came across MitoBright IM Red in our research. This one also seems to be designed for use in fixed cells, specifically for those immunostaining experiments.
Yes, MitoBright IM Red is another great option when you need to do immunostaining. It's specifically formulated to withstand all those washing and permeabilization steps that are part of the immunostaining protocol.
So it's like a really tough dye that can handle the rough treatment.
Yeah, you could say that. And the mechanism is similar to MitoTracker Red in that it forms those covalent bonds with proteins inside the mitochondria, making it very stable and resistant to washing out.
So in a nutshell, it allows researchers to visualize the shape of the mitochondria very reliably, even in fixed cells where they're also looking at other things.
Exactly. It's a great tool for those co-localization studies where you want to see how the mitochondria are positioned relative to other structures in the cell.
Now, are there any limitations or things to keep in mind when using MitoBright IM Red?
Well, because it's really designed for fixed cells, you can't use it to look at dynamic changes in mitochondrial morphology in living cells. And compared to MitoTracker Red, there's just less published research using MitoBright IM Red. So researchers might need to do a bit more optimization to make sure it works well in their particular experimental system.
Right. It's a relatively newer dye, so the scientific community is still building up that collective experience with it.
Exactly.
Okay, so we've talked about those 3 dyes in detail. Now let's circle back to some of the other dyes we mentioned earlier, like Rhodamine 123 and JC1. We categorize those mainly as membrane potential indicators, but how do they relate to morphology, even if that's not their primary purpose?
Yeah, so even though these dyes are primarily used to measure function, they can sometimes give us clues about morphology either directly or indirectly.
Okay, I'm curious. How does that work?
So take Rhodamine 123. We talked about how it can actually induce changes in mitochondrial shape.
Right, the dye itself can be a problem.
Exactly. But that also means that if you're using Rhodamine 123 and you see changes in morphology, you need to be very careful about interpreting those changes. Are they real or are they an artifact of the dye itself?
So it's like a cautionary tale, reminding us to be really critical of the data and to always consider the potential for the tool to influence what we're seeing.
Exactly. Now, JC1 is another interesting one. It's a ratiometric dye, meaning that its fluorescence changes color depending on the membrane potential.
So it can tell you how charged that mitochondrial battery is.
Right. And what's cool is that JC1 can sometimes reveal that different mitochondria within the same cell have different membrane potentials.
Ah, so some might be highly charged, others less so.
Exactly. And that often implies that those functionally different mitochondria, they might also have different morphologies.
So the ones with a higher membrane potential, the really active ones, might look different from the ones with a lower potential.
Exactly. It's not a direct measure of morphology, but it's a functional readout that can sometimes correlate with structural differences.
Okay, that's pretty neat. Function and form, always linked. What about TMRM and TMRE? They're also membrane potential indicators, right?
Yeah. TMRM and TMRE are often used to get very precise measurements of the membrane potential. And while their primary purpose isn't to visualize morphology, the way they accumulate in the mitochondria does outline the shape of the network.
So you can kind of see the shape, even if that's not the main thing you're looking at.
Exactly. But there's a caveat here. At high concentrations, these dyes can quench each other's fluorescence.
Quench. What does that mean?
It means that the fluorescence signal actually decreases if there's too much dye in one spot. So you might see a dimmer signal, not because there's less mitochondria, but because the dyes are interfering with each other.
Ah, so it could be misleading if you're not careful.
Right, so you have to be mindful of the concentration and be aware of this quenching effect.
Okay, so another layer of complexity to consider. Now, what about Sci5-PEG4? That one was mentioned as a newer dye, and it sounds pretty promising for certain types of research.
Yeah, Sci5-PEG4 is really exciting because it emits light in the near-infrared spectrum. And that makes it really useful for in vivo studies, particularly in the brain.
In vivo meaning in living organisms, right? As
Yeah.
opposed to cells in a dish.
Exactly. And the brain is a particularly challenging organ to image because it's encased in the skull and visible light doesn't penetrate very well.
So near-infrared light is like a superpower that lets you see deeper into tissues.
Exactly. It can penetrate further, and it also experiences less scattering, which means the images are clearer. And what makes Sci5-PEG4 even more exciting is that it can cross the blood-brain barrier.
That's the protective barrier that keeps a lot of things out of the brain, right?
Exactly. So it opens up the possibility of studying mitochondrial morphology in neurons in living animals, which is incredibly important for understanding neurological diseases.
Wow, that's huge. So a lot of potential there.
Definitely. But as with any newer tool, there's still a lot of work to be done in terms of characterizing its properties and optimizing its use.
Right, the research community is still figuring out all the nuances.
Absolutely.
Okay, last but not least, we have Fura-FF. This one's a calcium indicator, so how does it fit into our discussion of mitochondrial morphology?
So Fura-FF is primarily used to measure calcium levels inside cells. And we know that mitochondria play a role in calcium signaling.
Right, they can take up and release calcium ions.
Exactly. So while Fura-FF can localize to mitochondria, it's not really the ideal tool for detailed morphology studies.
As it's not specifically designed for that purpose.
Right. There are other dyes that are much better suited for visualizing the intricate shape of the mitochondria.
So it might give you a general idea of where the mitochondria are, but if you really want to look at their structure, you'd use a different dye.
Exactly.
All right, so we've covered a lot of ground here, talking about all these different dyes and how they work. But once researchers have captured those beautiful fluorescent images of mitochondria, how do they actually go about analyzing that morphology? It can't just be a matter of looking at the pictures and saying, oh, those mitochondria look kind of fragmented, right?
Oh, absolutely not. It's way more sophisticated than that. There's a whole field of quantitative analysis of microscopy images. Researchers use all kinds of specialized software tools to extract meaningful data from those pictures.
So it's not just subjective eyeball assessments.
Yep. It's all about numbers, measurements, statistics, and there are some really amazing software programs out there that can do some pretty incredible things.
Okay, so give us some examples. What kinds of software are we talking about?
There are open source platforms like ImageJ, which is super popular, and it has all these plugins specifically designed for mitochondrial analysis, like MiA, Mitochondria Analyzer, and MitoGraph.
So those are free tools that anyone can use.
Exactly. And then there are commercial software packages that have even more bells and whistles, like Amaris, Amira, Cell Profiler. There's even specialized software that doesn't require any labeling at all, like NanoLive. And then there's MITR, which is specifically designed for analyzing mitochondrial networks.
Wow, it sounds like a whole toolbox of options.
Oh, it is. And each one has its strengths and weaknesses, so researchers choose the one that's best suited for their particular research question.
So what kinds of things can these software programs actually do with those mitochondrial images?
Oh, all sorts of things. For example, they can calculate the area of individual mitochondria, their perimeter, how circular they are, which can tell you if they're fragmented or elongated. They can measure the aspect ratio, which is the ratio of length to width.
So they're putting numbers to those visual observations of fragmentation versus elongation.
Exactly. And that's really important because those changes in morphology, they're not just cosmetic. They can have real functional consequences for the cell. Like fragmented mitochondria are often associated with stress, disease, aging, while elongated, interconnected mitochondria are generally seen as a sign of health and efficient energy production.
Right, we talked about that earlier. So these software programs are letting researchers quantify those changes in a really precise way. What other kinds of analyses can they do?
Well, they can also do things like network analysis, looking at how interconnected the mitochondria are within the cell. They can measure the number of branches, the length of those branches, how much they're fusing and dividing.
So they're really getting into the nitty-gritty of that mitochondrial network structure.
Exactly. And if you have time-lapse images, like movies of the mitochondria over time, these programs can track their movement, their interactions with other organelles, even individual fusion and fission events.
Wow, it's like they're giving the researchers a whole new level of insight into the dynamic world of these organelles.
Absolutely. And it's all thanks to these powerful computational tools.
All right, so before we wrap up, we need to talk about one more approach to visualizing mitochondria, and that's using genetically encoded indicators, or GEIs.
Yes, GEIs are a really cool alternative to using those synthetic dyes.
So how do they work?
Well, instead of adding a dye to the cells, you basically engineer the cells to produce their own fluorescent marker that's specifically targeted to the mitochondria.
So it's like the mitochondria themselves are glowing from the inside out.
Exactly. It's usually done using a gene for green fluorescent protein, GFP, which is attached to a specific sequence that tells it to go to the mitochondria.
So it's like a molecular zip code that directs the protein to the right place.
Exactly. And this approach has some advantages. For example, because the fluorescent protein is constantly being produced by the cell, it's great for long-term studies. You don't have to worry about the dye fading or washing out.
So you can track those mitochondria over long periods.
Exactly. And it's also very specific, for sure that you're only labeling the mitochondria. But there can be some downsides too. Like the production of the protein can be slower than the uptake of a dye. And sometimes overexpressing a protein can stress the cell.
So there are trade-offs to consider, just like with the dyes.
Absolutely.
Yeah.
But overall, GEIs are a really valuable tool for certain types of experiments, particularly when you need long-term labeling or very specific targeting.
Okay, so we've covered a lot of ground today. We've talked about the challenges of using fluorescent dyes to study mitochondrial morphology. We've delved into the mechanisms of specific dyes, and we've even touched on those powerful software tools that help researchers analyze the images.
It's a lot to take in, but hopefully it's given our listeners a deeper appreciation for the complexity and the beauty of this research.
Absolutely. And before we say goodbye, I think it's worth leaving our listeners with a thought to ponder.
Always a good idea.
So we've talked about how these dyes can sometimes interact with mitochondrial function and how the choice of dye can influence what we see. So the question is, how much does the choice of dye and its potential to subtly alter mitochondrial behavior actually shape our understanding of cellular processes?
That's a really great question. It's something that researchers are constantly grappling with, trying to tease apart what's a real biological phenomenon and what might be an artifact of the tool itself.
Right. It's a reminder that even with the most sophisticated tools, we always have to be critical and aware of potential biases.
Absolutely. And as we continue to develop new and even more advanced tools for visualizing mitochondria, that awareness is going to become even more important.
Well, on that note, we'll leave it there for today. Thank you all for joining us on this deep dive into the world of mitochondrial morphology. As always, we encourage you to explore further, dig into those research papers, and keep those questions coming.
And who knows, maybe one of our listeners will be the one to develop the next groundbreaking tool for visualizing these fascinating organelles.
Absolutely. Until next time, happy exploring.